Hi, you guys. I want to show you how to look at our data a little bit. We have some sequences back, and I put them on the public drive and also in Dropbox. So I'm going to show you what we want to do is first we have 4 different sequence files for each plasmid. So if I go to the public drive, or actually I'm not going to go to the public drive because I have them in a different place than you. But if you guys go to the public drive, then you should be able to see a folder here, sequence data. If you click on that, open it up, and you see all of these sequence files. And there's all different types of files. And we won't have time to kind of get into a lot of what these are. Some of these allow you to go back and check the sequence quality and throw out bad data points if you want. What we're interested in is scrolling over or down to these .seq files, .sequence files. And those are the ones that we're interested in. And you'll notice that there's 4 of them for each plasmid, and I named them with people's initials. These are Collins, Juan's, and then these are group ones, Laura, Deanna, and Shay. And then it has lomatium or cilantro or Arabidopsis, and then the PCR well that they originally came from, and then the primers that were used to sequence them. And so if you remember, all these primers allowed us to get overlapping sets of sequences. So what we have are partial sequences that none of them individually covers the entire GAPDH insert in the plasmid. But together, if you could put them all together somehow, they would cover that entire region. And so that's what I want to show you now, is how to put them all together and get one continuous sequence. So the way we're going to do that is using an online program called CAP3. And I'll show you how to find it. If you Google CAP3, it should be the first, I don't know, 100,000 hits that come up. There's a lot of different servers that run the program. NCBI hosts a version of it. This one's very easy to use. The very first one that comes up here on the doua.prabi.france site. So I'm just going to go to that one. And you can see here it's got a field that lets you enter FASTA files. And so what we're going to do is we're going to enter all 4 of our FASTA files for each plasmid that we're sequencing, and this will assemble those sequences for us. And so what you can do is you can go back and open up these files, and you can see this one actually doesn't end up having a lot of data. N means it couldn't determine what the base pair was at that location. But this one has a lot more sequence, and you can actually see here's that little caret that we know is the signifier starting the FASTA file. And as you scroll to the right, this is just a plain text file, so I opened it in Notepad. And you'll want to probably use Notepad or a text editing program to do this. You can scroll to the right and you can see all this sequence file. You can just hit Control-A or Edit, select all, and then copy, right? Edit, copy, and then you go back over to CAP3 and you plop it in. So if you scroll up now, you can see the beginning of our FASTA file all the way down to the end. And I'm not going to click through all of them here to show you. This is going to be like a cooking show. I have one all set up to go. So you do that for each one and enter all of them together. And so I'm just going to do that now and paste them all in at once. So if I put all of those in together, then you should be able to scroll down it now and see the gap sequence forward primer, and you scroll down, and that one ends, and the next one starts gap sequence reverse primer, and you scroll down, and that one ends, and the next one, the PJet sequence forward primer that obtained that sequence read, and then the PJet sequence reverse primer that obtained that sequence read. And so now I've got 4 sequences in that represents one plasmid that we sequenced. If you hit submit, it gives you some options here. The first one is what you want, contigs, and it says contig 1 or continuous sequence 1. It took all of those sequences, overlapped the parts that overlapped, and it also reverse complemented the reverse sequences. It figured out that those were going in the other direction and found where those would hit. And so this contig 1 sequence is now your sequence plasmid from start to end. The best thing to do with this sequence is probably to save it in a separate file just for your own records, maybe just a notepad file or a Word file so that you have it for your records.